I want to welcome everyone. You're in the right place. This is our virtual roundtable on translating data science. And today we're going to talk about bias in data sets and analytics. And if you can hear a siren in the background, that means it's noon on Wednesday, and in my town, they test the fire system at this moment. I want to introduce Michael Welling. My name is Joanne Geer. I'm the Executive Director for the Westchester Biotech Project. I want to introduce Michael Welling, who will share a little bit about our organization.
Thank you, Joanne, and good afternoon, everyone. Thank you for joining us today in what we know will be another sort of insightful and exciting conversation, bringing together different aspects of our community. The Westchester Biotech Project is a nonprofit organization whose sort of main core mission is to bring together researchers, data scientists, and engineers to foster collaboration, convene on solving problems, and hopefully help further everyone's interest, research, and projects for which they've undertaken. We launched it's almost 2 years ago. We're not quite 2 years old yet. And since that time, our organization has had the opportunity to engage with folks from around the region, around the country, and around the globe. You'll see today from our speakers, we have folks from almost all corners of our country. And you may have seen, if you're on our newsletter, last week we announced our MOU that we signed with Westchester Biotech Project Europe and a sister city project in France, where we believe we're going to open up a corridor for sharing of ideas and introducing of companies and opportunities, sort of Atlantically, if that's a word. As you see on this slide here, some of our other efforts that we have underway today, obviously, is our translating of data science. You see that we have our consortium on the microbiome. We have our rare disease efforts. We have our young investigators. We have a number of initiatives underway. And I would encourage all of you, if you haven't already, or if you haven't in some time, to poke around our website and see some of the events and activities that are coming up. And we've got some really, our next symposium is on March 27th, Innovation and Research 3. And we've already got some fascinating speakers signed up and would encourage everyone to participate. As always, the work that we do wouldn't be possible without the support of our community partners, alliance partners, and our participants. Obviously, the screen that's before you here, it would take most of our time today if I were to mention and talk about every single name that's on here. But one of the things that I always like to say is that when you are out there and you're conducting your business, we encourage you to engage our partners who support us as we make every effort to support them whenever possible. Next slide. Am I handing this over? Am I still going, Joanne?
I think if you want to finish, if there's anything more you want to say, now's the time to say it, and then we'll move on and invite Paul to say a few words.
I thought I started this organization so I could say anything I wanted at any time, but I think I will turn this over to Paul now. No, let me let it turn over to Paul, and so we can jump into this.
Oh, great. Well, so this roundtable is chaired by Paul Savage and Antonio Biancardi, and we're just going to hear a moment from them. Paul, would you like to say a few words?
Yes. Good morning, everyone, and thank you for joining us. This is a wonderful opportunity, and I would certainly like to applaud the subjects that have been coming out of the association. This is a tremendous opportunity, and certainly it's quite current and relevant to be focusing on the quality of data sets that we potentially work with. In my own particular case, I've been working with massive healthcare databases measuring 200 million patient records. And these numbers are growing exponentially. In the next year, we'll probably have 100 times as many records to work with. And trying to get down to the quality of healthcare is a tremendous opportunity for us to leverage the data environment. So I again applaud everyone's participation and my appreciation. Thank you.
Thank you, Paul. Antonio?
Hello, everybody. My name is Antonio Biancardi. I'm a board member at the Westchester Biotech Project. I'm very proud of that fact because I love what the organization is up to. And I have to say kudos to you, Michael and Joanne, for the effort that you've put in, and some of the initiatives that you're running are just amazing. So a little bit about me. I've been building software for the last 20 years, and one of the things I noticed early on is that there's a big disconnect between the people who are working with the technology and building it, and the people who are actually using the technology, which led me to start thinking about the world of data science and its application and the problems that are inherent within just the dynamic between the people working with the technology and the people who are ultimately going to use it. And in the area of data science, specifically in the world of life sciences, there's just tremendous value to humanity here. So I'm really happy to be a part of this initiative because I believe that if we can start to bridge that gap, start to inform technologists about the opportunity and really start to inform researchers about really just ways of using the tools and start to build better tools, I think that collectively we can make a huge impact on the world. And so I'm really proud to be a part of this, and I thank you all for joining us today. And as Paul said, you know, the initiative is really taking on a life of its own, and we're going into some really very interesting areas. I see tremendous potential for this, and I'm just really happy to be a part of this. Thank you all.
Thank you, Antonio. So we're going to hear from several different folks, and I'm going to, in Hareesh, in a moment, I'm going to introduce you and give you the control of your slides. I just would like to mention that this particular roundtable, it has 2 additional events planned this spring. On April 23rd, we're going to be doing a roundtable on data ownership and security and privacy. And on May 14th, we're combining with our consortium on translational research in the microbiome and to discuss the development of a research data infrastructure development to focus on the microbiome. So I'm excited about combining the streams there. Let's see, and Hareesh, I'm going to make you, so we're going to introduce Hareesh Chandrupatla, and I'm going to make you the presenter, and I got to find you on my list. Here you are. And you can unmute yourself and tell us about yourself and show your slides. You should have a pop-up on your screen now. I think you're muted if you're talking. Hareesh. Okay, I'm not sure what the issue is. I will, there you go. I'll just let everyone know that we're going to have all of our presenters, and then we're actually going to invite everybody on the call to introduce yourself and talk about what you see here and ask questions. So here we go. And there you go. Going full screen. Yep. Beautiful.
Okay. All right. Well, my name is Hareesh Chandrupatla. I'm the founder, chief data scientist at Anjin Analytics. We're a technology consulting company focused on advanced analytics, big data, AI for healthcare and life science. So thanks for the opportunity to present. Just here on topic of bias in data sets. I'm really just have a few slides, just giving some examples maybe we've seen in the news and a use case that I reflected on in the last webinar, where there was, a really good example of bias, and just wanted to bring that up as, use case. So recently in the news, may have heard about Amazon. Well, Amazon is in the news for a lot of different reasons, but in terms of bias, they developed a recruiting tool, which they've was showing bias against women. The system taught itself that male candidates were preferable, and specifically, anywhere with gender, especially with, so if a woman went to a certain women's college or it's a women's glee club, it was finding that the system was just creating bias against women, and so how does the system just do that? I mean, using like unsupervised methods or a self-learning system that isn't monitored by humans, we can see, system learning to create bias that does not reflect the reality of how we want these systems to make decisions. And so there's some issues here with selection bias. So is the data set representative, and are the decisions representative of what we desire? So I think it's a good selection. Selection bias is something here that has come up. Confirmation bias, maybe the system itself, once it decides, okay, a woman's not a preferred candidate, it can then back it up by falsely confirming that, this is based on, valid results. So I think, systems like this require humans in the loop to continually monitor and manage the system, and inherently the algorithm itself needs to take into account, the bias that we do not want it to reflect. Another example.
Hareesh, I'm sorry, we're having an issue of seeing your slides. So I'm going to take the screen for a moment, and then I'm going to give it back to you. I think we're following along with your comments, but it certainly would be nice to see your slides. So I'm going to show my screen for a moment. I'm going to try anyway.
Yeah, we can see Hareesh's now. Just when you clicked full screen, we lost you, so something happened.
Okay, so you can see the screen right now?
Yeah.
Thanks, Chris.
Okay.
I'm actually not seeing the slide, so something happened on my screen, but we're going to be fine.
All right, so is this fine? I don't have to put it into full screen.
That's fine.
Okay.
Thanks.
Just another example in the news. A number of courts around the country use software that provide risk scores that help judges make decisions on sentencing. So there's around 137 questions that are asked in the system, and while they don't directly incorporate the question of what is the race of the individual, a lot of the questions are looking at things like salary, the family situation, did the person grow up divorced, in a divorced family, a lot of different social determinants questions, as well as location, where they live. And so a lot of these questions, even though they don't directly ask concern race, they may indicate a bias towards race. And so they're finding that these systems have a lot of higher rate of false positives for African Americans. And so, it's just bringing up another issue where, decisions are being made by judges to provide sentencing for people. And if the software, is not being fair and balanced, therefore our sentencing is not going to be fair and balanced as well. So this is, I think, another major concern that needs to be addressed. And so, moving on just to a use case for a project that we worked on for a startup that was focusing on wound care clinical decision support. Just to give the case, we were developing a software that physicians could use to monitor chronic wounds for patients who had diabetes and other chronic issues. And so the goal is to develop a software that could automate the detection of the perimeter and boundary and area of a wound from an image. So just by taking a picture of a wound, being able to detect, these features was our goal. And in order to get this started, we were provided by one of our partners with over 20,000 images of chronic wounds. And we found in the data set that about 70% were Caucasian, 20% were African American, 10% South Asian. And just to give these statistics around the population. And the methodology we were using, convolutional neural networks. And really the method fundamentally works by looking at the difference in the skin pigmentation between the healthy and the wounded skin to kind of to develop its to develop the algorithm and understand what to try to predict a boundary. So what we found was that we couldn't just create a training set with a mix of all of these populations because of the drastic differences between the healthy skin for different populations versus the color of the wound. And so what we found was we had to separate out the population. So for Caucasian, we had to work on algorithm development for that population, and then separate out African American and separate out South Asian population. And as a result, because we have so many more Caucasians in the population, we're going to have higher performing predictions. So that creates a bit of selection bias. So anytime a new patient comes to the system, if there's a Caucasian, we're going to have a better results because we've been able to train on a larger data set. And so inherently the system, comes biased towards, the training data we have available. And so we have to be sensitive to that when we, provide results. And so over time, we have to make note of that and ensure that, as we, as more new patients come to the system, make sure we can track and use, increase the number of African Americans, increase the number of South Asians and other populations so that we can properly reflect the reality of our populations that are coming through the system. But this is, I think, one major issue with, let's say, healthcare data is any algorithm that's developed is going to be reflective of the population and data that's available for, a given hospital or a given area. It's going to be biased in different ways. And so the initial algorithm that's developed here with these 20,000 images, we may not just be able to take it to another country or another hospital system. We'll have to make sure that we can get new images, new data from each location or new client to kind of ensure that we get more diversity and also reflect the population that we're working with. So those are just a few slides here. That's comments I had to make about bias in a few different use cases. Thanks.
Well, thank you. Thank you very much. We're going to now hear from Scott Lett. I'm going to make you the presenter, Scott.
Okay, are we good?
For some reason, I'm not seeing any slides. Is everybody else seeing Scott's slides?
Yeah. Looks beautiful.
Yep.
Good. Okay, great.
Okay, so my name is Scott Lett. I'm currently working as a machine learning and data science consultant out of Colorado for a company that is just me called the Holistic Mathematics Agency that I've actually had for close to 25 years. I've worked though with a bunch of large and small companies, everything from startups to large multinational companies in various areas that you can see here. One of the things that has changed over time is moving to bigger and bigger data, moving away from knowledge-based models to data-driven models, and also models that more and more affect a population of people rather than just an individual engineered system. So I thought that first talk was awesome in terms of talking about how the technical aspects of modeling interact with the fairness aspects and how bias shows up in both places. So I thought I'd talk a little bit about the traditional kind of bias and then jump a little bit into the fairness types of bias and throw some hand grenades into ideas for what could be done about that when fostering using data science on big data sets. One of the things that I've noticed recently is that more and more people that are doing their business pitches with their technologies is that they're being asked about the risk side of their business plan and whether they've addressed it. And one of the risks that they're asked about when it involves medical data or private data is whether they've planned for securing the data, the private data. One of the things that's not being asked yet, but I expect it to happen over time, is the risk to the business that accrues to building unfairness into their system and what liabilities might be incurred because of that. So stay tuned. The traditional types of bias are either statistical or cognitive, basically. There's sample bias, and I think that we already saw an example of that where sometimes it's easier to get data from one population that tends to be a privileged population and harder to get data from other groups, which means that your results are going to tend to be better for that population. Prejudice bias isn't necessarily the prejudice of the modeler, but it can be. There's measurement bias, and that is over time, we use different assays and different modalities for trying to measure data from especially patients. And as those modalities change, it makes it harder and harder to combine data sets because they look like they might be measuring the same thing, but because the protocols are different or the technologies are different, we have biases in the measurements themselves that can create errors in the predictions. And then there's algorithm bias. And I'll talk a little bit about that later, but basically if an algorithm consistently underestimates or overestimates some quantity, then that builds an error into the system that wasn't possibly in the original data. So one of the traditional sources of sample bias is that clinical trials, when you're trying to launch a new idea or a drug or a diagnostic, is that clinical trials are expensive and the budgets are limited, and so sometimes you go for the easiest data to get. And one way that we've done to sort of mitigate that is to do what's called meta-analysis, and that's where you pool data from multiple studies so that you get improved results from the combination of data. One of my most referenced studies was an effort we did with the University of Cincinnati to study the protein called osteopontin, which has since that study become very actively researched in terms of using it to have a screening test or an assay that predicts whether somebody not only has cancer, but is it a type of cancer that needs treatment. Publication bias, though, creeps into meta-studies, and I wanted to talk a little bit about that. And publication bias occurs when people that get a result that's kind of uninteresting, that didn't result in something new, like overturning a previous known result, that they tend not to publish those results. And so studies with significant results have been found to be 3 times more likely to be published than studies with negative results. Over to the right, you can see some things we did to try to detect and even mitigate publication bias. And I don't get too technical in the talk, but if you look at the top funnel plot pictures, what you see across the bottom are over a number of different studies. The different studies measured an effect size. In this case, how aggressive was a cancer? And the vertical axis is the sample size of each study. And what you expect to see is that all of the studies will tend to cluster around what's the true effect of whatever the thing is you're trying to measure. And there will be, as the studies are smaller and smaller, that they can be less accurate or less precise, and they can drift farther away. And you can see here that some of the studies actually show a negative effect size or near zero. And that's what you would like to see if in fact people are not censoring their own publications, or if the journals are not censoring them. If this plot were less triangular in shape around the main effect size, you would not see studies with effect sizes near zero, that they would tend to be sort of skewed. We do see these two studies that were very small that showed a really big effect size, but we found that was not significant. The plot below that is another way of looking at the same information, and some different statistical tests to try to detect whether the pooled data actually has some missing information. Confounding variables are a notorious way to get the wrong result from data. And even if it's one of those things where just adding more data doesn't fix the problem. An example from work we did was cardiac drug safety. And the surrogate for that in the preclinical development of drugs has always been to look at the action of drugs on what's called the IKR sodium channel. Now, to explain a little bit more about that, the schematic diagram at the bottom is sort of a graphical representation of a heart cell. And it could be a muscle cell or a Purkinje cell. And all of those little arrowed things represent channels that are proteins that go across the membrane of cells through which different ions can travel. And when a cardiac cell experiences a difference in electrical voltage between the inside and the outside of the cell, then some of those channels will open and start moving ions into or out of the cell, and creating a voltage difference. And when that voltage difference gets big enough, then the muscle cell contracts, and then the cell resets itself back to the original resting state, and we move on. And things like cardiac arrhythmias and tachycardia and fibrillation are basically malfunctions of this system. And some of those malfunctions can be caused by drugs, and almost every drug or every class of drug hits the IKR channel, which I think is down at the bottom somewhere. Yes, it's this little guy back here. And traditionally that was used as the screen. That was what they knew about to screen for. Many drugs bind to that protein and affect the rate at which the cells recover from a heartbeat. And the problem with that is that most of the data is available from a certain group of people, and if you just use the sort of that surrogate to determine what the safe doses are for drugs and whether a drug is safe, then what you'll find is that you are preferentially getting a better result for white men and getting worse results for women, for people of African descent, for people of Southeast Asian descent, and people with various kinds of health and genetic risk factors. So what we did was we said, well, drug industry, if you measure the effect of your drugs on all of these things, we can tell you with our models what the drugs will do in cardiac safety sense. And they said, yeah, that's one more thing we're not going to do. That would cost us zillions of dollars, and it would tell us things we don't want to know about our drugs. So what we did was we said, well, we'll look at the data you're getting. The data on the left are data that's measured in a laboratory on animal heart cells. And basically it's the electrical voltage between the inside and the outside of those cells, and over time under different doses of a drug. And if the drug is a traditional one that causes a certain kind of fibrillation, then you'll see this sort of as increasing dose, you'll see this recovery wave of this voltage stretch out farther and farther in time. And so the top row represents data for a particular drug known to cause fibrillation in some patients. What we were presented with was data for some compounds that were in development at a drug company that didn't show the same sort of monotone increasing behavior, and they didn't know why. So what we did was we used machine learning, and we found that we could infer the effect of those drugs on some of those channels in the previous picture, and then use that information to then model the EKG of a variety of different patients under different risk conditions and different doses, and predict which drugs had a risk for cardiac drug safety. We had a lot of pushback from experts because some of those channels that we pointed to that were being affected by these drugs, the NIH reviewers, for example, declined to fund our idea because the reviewers said that some of those channels, the sodium channel in particular, was not important, and so our proposal was declined, and we moved on. But what was found out later was, in fact, it's now standard knowledge that that channel is very important, and some of the genetic variations and some of the drug effects are very key in predicting drug safety. There's also prejudice bias, and that is, was mentioned before, that if you get a result from your model or from your data that you want, that, for example, can get your idea funded or make a lot of money, then you tend to stop looking for additional results, or stop looking for data that can disprove your model. And so that's a very famous one that also involved confounding variables, was a study all the way back in 1896 by Hoffman for the Prudential Insurance Company, in which he determined with a lengthy analysis that Black people had such precarious lives that they could not be insured, and that preferentially harmed Black people in terms of insurability for decades after that. But what he didn't look at was what was the effect of income and injustice on his model. And if he took those things into account, then he would find that white people in the same conditions were having the same precarious lives. Availability bias is one of the ones we sort of talked about, and that is we tend to make quick decisions based on the immediately available information, especially if it confirms our bias. And these two things together have been listed as some of the main causes of the reproducibility problem in science. A study by Anjin on published papers found that the majority of the papers they considered had completely unreproducible results. And it's thought that two things contributed. These two things and the incentives that people have contributed to unreproducible results. Measurement bias, I talked about those already, so I'll sort of skip over that. The algorithm bias, here's my simple result that doesn't have anything to do with medicine. If I have a hat in which I place some cards that are numbered 1 through n, and I draw one out at random, and the challenge is to estimate how many cards there are in the hat. Well, it turns out that maximum likelihood, which is a popular statistical machine learning method, will always underestimate by almost a factor of 2 the number of cards in the hat. And so in this case, if you pick out a card that's numbered, for example, 6, then the maximum likelihood estimate for the number of cards in the hat is 6. But in fact, the unbiased answer is 11. And if you understand how your algorithm biases your answers, it is possible to use a biased algorithm and correct it afterwards with post-processing. So non-traditional bias that we've already touched upon. Purpose bias, which is sometimes we think that the purpose of a model is one thing, but it's actually something else. So for example, we think of the modeling that's done in the insurance industry as trying to find people with certain risks and try to group them together or assign their pricing based on the amount of risk. But in fact, a lot of purposes of models that have been used in the insurance industry have been found to be instead identifying unprivileged groups that had few choices for their insurance and to charge them more because essentially they could be charged monopoly prices. Another kind of purpose bias is fraud, identifying susceptible groups so that they can be targeted for fraudulent activities. And I think a lot of those have been showing up in the news, so I won't touch on them too much now. But those are two things that I don't think we're trying to do here, but they show up in sort of this non-traditional bias. Access bias. If decisions that affect me use these really advanced models, then it's really difficult for me to even know that a model is being used, let alone understand why that model predicted, for example, that I shouldn't get health insurance, or I should be charged twice as much, or that I should be locked in jail when someone else with a similar background is not locked in jail. The whole thing is a black box and so complicated that it's difficult to find out exactly why these decisions are made. And we think of these models as being, sort of objective, but in fact they're not. And so I'm inspired in a lot of this discussion by stories in the news and also by the book Cathy O'Neill's Weapons of Mass Destruction, which makes a great read. Model bias can show up when, in fact, for various reasons, and when a false positive rate, for example, for a diagnostic test or predicting somebody is likely to commit a crime is worse for an unprivileged group than it is for a privileged group, then the model itself can introduce a bias into the problem. And so there's a set of metrics and literature now appearing around fairness, both in data sets and in analytics, that I'm going to talk a little bit about, but just touch on it just to start up arguments. So one of the things that's come out recently that I thought I would throw out there is that IBM has made available to open source a set of tools in a package called Artificial Intelligence Fairness 360. It's sort of an amazing effort, but what thing it does for me is sort of organize thoughts around what's being done to measure fairness, build that measurement into modeling and data analysis, and improve fairness. And so I thought I'd talk a little bit about what this package can do. It provides fairness metrics that can be used on your data sets or your models, and it provides algorithms that are mitigations that help correct biases. So in terms of dividing up what it does, part of it divides up into the model training data. What training data is used to teach machine learning algorithms what it is they need to predict. And in enhancing this data to have some new attributes in terms of, instead of just saying, well, I've got a group of people who have a certain age, for example, I may divide that group up into people who have traditionally in a particular context been privileged or unprivileged. For example, people below a certain age may be unprivileged, and people above a certain age may be privileged, or vice versa. People that are older or younger might be privileged. Sex might be an attribute that can be labeled as with attributes of privileged. In addition, some of these attributes are annotated with protected, meaning it's a quantity that we would like to predict in our models that we would prefer be equal for different groups, regardless of whether they're privileged or unprivileged. So in addition, the system provides metrics for how fair the data or the models are. And those are really interesting, and I'm not going to go too much into them here, just to say that the tutorials that are available for them provide a lot of interesting information that's accessible to non-technology people. And then there are mitigation algorithms that can be used either in pre-processing the data, during the training of machine learning algorithms, or in correcting the results in post-processing. And then I find this is really important. One of the biases I think about machine learning models is that some people know why they're giving a certain answer and other people don't. And what these systems do is actually explains their results in terms of what was causing unfairness in the system and what was done to correct it. And then finally, a lot of industry-specific tutorials and guidance are provided. So I think for people that are interested, it's a fun place to look for what research is being done and what practical tools are being made available in this space. So my thoughts on this are that a group like this that's involved with either launching new ideas or facilitating them through providing data or funding or teaching students, that guidance in the area of both the technical and fairness areas of bias is really important. You know, how to see it, how to catch yourself when you're committing some of these cognitive biases in your modeling, and also how to correct the problems. The tools, making them available, even contributing new tools, I think is a really potential fruitful area for universities and research groups to be doing. Data, of course, pooling data, labeling them in terms of what are traditionally some privileged and unprivileged groups, what things might correlate to privileged or unprivileged groups, and also fairness assessments of either the data or outcome of models that are created from that data. And of course, funding to make sure that people have the additional resources needed to make their models work better. And then finally, looking at the incentives, you know, what is it about getting to market that provides a disincentive to making your models better, and what could be done to improve that? I think are areas that are interesting and important for how to make things better for the future. And with that, I'm going to shut up and say thanks.
Thank you, Scott. That's fantastic. I'm going to show my screen now, and we're going to move to Cruz Street. Let's see. I'm having a little glitch at my end. Let's see if I can do it this way.
Yeah, my street's completely unpronounceable. It's Kinnikinnick Court. So.
All right. Well, for some reason my system does not want to share my screen. I don't know why. So I'm going to you guys at Cruz Street, who's got the best access to your slides, and I'll make you the presenter.
Yep. No problem. Pass it to Jesse. Be fine.
That's Jesse. Okay. Let's see. Here you are. And there you go. You should have a little pop-up.
Tell me if you can see my screen.
Can folks see Jesse's screen?
Yep.
Yes.
Yes.
Okay.
Great.
Yes.
Beautiful. Take it away.
All right. Thank you. First of all, thank you so much, guys, for spending some time with me today. I am super excited to share this time with you guys and really enjoying the presentations I've heard so far. I wanted to just share with you a little bit about Cruz Street. Cruz Street is a company that's sort of at the intersection of data science and business. Our goal really is to push forward as a mission to accelerate the business community's adoption of data science for the purposes of problem-solving growth, and hopefully giving people insight into something that is a little bit overly complicated for a lot of businesses to simplify. So we're really trying to simplify it for us. The folks who are presenting today, briefly, I'll kind of introduce. First off, Yacine's going to be talking a bit about data quality. Yacine is Vice President of Data Services at Cruz Street. He actually comes with a wealth of experience, decades of experience working for some of the largest big data companies in the world. Most recently at Experian, he was managing one of the largest data sets, consumer databases in the world. Also has background in intellectual property creation with 2 patents in data anonymization. And Jim Duarte is actually a consultant advisor for Cruz Street, also data scientist, and owns his own firm, LJ Duarte and Associates. Jim actually has a upcoming series he's publishing in Forbes, so if anybody's interested, follow up with him after. He has 6 patents in analytical methods and has worked for SAS, as well as some big brands like Anheuser-Busch. So with that, why don't we go ahead and dive into healthcare data quality impact on bias. Yacine, take it away. Yacine, are you there? Okay, maybe, can anybody hear me? I want to make sure I'm not...
Yeah, we can hear you.
Yeah, okay. Yacine, I think you might be on mute. Okay, why don't we go ahead? I'm going to do this. I'm going to let Jim start, and we'll come back to Yacine, unless he can get on here. Yeah, we'll let Jim start. Okay. So Jim, whenever you're ready, take it away.
Okay, the previous talk's been very, very interesting, and I enjoy that. One of the things that I got into in doing
hello?
Healthcare.
Ah, there you are.
Oh, I was talking
right.
to myself. Looked like I was talking to myself for a few minutes there. Okay, good afternoon, everyone. My name is Yacine Abdous. I'm VP of Data Services with Cruz Street. I'm going to talk about data quality and the downstream consequences of bad data on healthcare analytics. Fundamentally, data used for healthcare must be of high quality to ensure that the information generated from analytics is valid and useful, well documented, to be used in context with the correct meaning, and easily accessible, for instance, available in a data warehouse or a data lake. The most important aspect of any analytics system is access to high quality data that is accurate, complete, timely, and relevant. Before any analysis is performed, ensuring that the source data is trustworthy must be of the top priority to any organization, especially healthcare organizations. Without data that is accurate, complete, timely, and relevant, it is impossible to trust in the results of analytics, and processing using that data would be suspect at most. Poor data quality and the lack of understanding the data creates huge challenges in healthcare. That is compounded by the fact that the highest priority initiatives for HCOs, healthcare organizations have today are mostly data driven. Healthcare research and
Hi, Yacine. We've lost you again. We may have a network issue.
downstream. Oh,
Okay, we hear
really?
you. We hear you now. So if you just repeat the
Okay,
last couple sentences that you said, we should follow up with you.
Said understanding the data creates huge challenges in healthcare. That is compounded by the fact that the highest priority initiatives that healthcare organizations have today are mostly data driven. Healthcare research and analytics initiatives have been hindered by data quality issues. Bad data causes bad outcomes downstream, such as lack of insight and analytical bias, poor healthcare organizations, financial and clinical decisions, and also poor system integration. Between the different systems in a healthcare organization. Now, causes of bad data quality needs to be identified. Addressing these root causes of poor data quality would be greatly enhancing the quality of data that is available for analytics. Common gaps listed here on this slide are mainly the root causes to blame for most of the data quality issues encountered in healthcare organizations. For example, software issues such as poorly designed data capture forms or data validation, and also coding standards and lack of metadata and lineage introduce that data quality issues. Beside putting in place a data governance that will help fix the data quality issues in healthcare across the data lifecycle, we can use data quality tools and processes to profile, discover, clean, standardize, validate. We can also enrich records with other data sources that can improve analytics. We need to put in place monitoring tools also to control the data quality over time and assess the quality of the data in its different complete. Can you go to slide to this, the next slide? Hello?
Yes, I've moved to the next slide.
Okay. Here, we can see healthcare information systems are generating huge volumes of data. As the growth rate of healthcare IT adoption continues to increase, the volume of data collected by these systems will exponentially increase also. As we can see here in this slide, data would be coming from different sources and stored on different systems. The same customer information is often stored on siloed systems and duplicated in different data stores. This will potentially create data quality issues that will affect analytics. While this very large and growing volumes of data presents an exciting potential for using analytics in healthcare, it is not a trivial task to ensure that this data is available and usable for this purpose. I must add that implementing a solid data governance is of utmost importance to alleviate data quality issues. Now, I'll give the floor to Jim, who will give more details about the impact of data quality on bias.
Thank you, Yacine. In the previous talks, we've heard a lot about building models and things like that. And traditionally, in the education of healthcare professionals, they get biostatistics training in what I call research biostatistics. They learn to look at clinical studies, hypothesis tests, and comparing p-values, and that's the big basis for most of the training for healthcare professionals. As I was doing consulting with the Veterans Administration, we came across the issue of looking at process improvement. So I'm going to take a different tact here and look at data through time or process statistics. So looking at data through time is a different type of process than looking at comparing groups or looking at certain groupings of data. So we're looking at things like signal to noise or random variation. How do we find a signal? If you're sitting in an operating room, you know that people are watching monitors for the patient, and they're looking for signals on any changes in the patient's condition or things that are going on. We can do the same thing for processes in systems, and I'm going to give you a few examples of where bias can come into a process when we do that, and then being able to look at those signals for root cause. And we'll see this on the next slide. I'll give you a couple of examples. Here we had a system in a medical center where we're looking at the dispensing of medications and looking at medication errors. And I like to take real data from an organization when I do the training of taking folks from their knowledge of research statistics and helping them with their knowledge in process statistics. Just as an aside, I have a pacemaker, and my cardiologist is always asking me questions about my lifestyle. I've been doing a lot of traveling and lecturing, and he asked me what I'm lecturing on, and I told him these topics and the work I'm doing with the Westchester Biotech, and he said, "Oh, that's interesting, the difference between research statistics and process statistics. Would you like to come and make a presentation to my group of cardiologists, because I think they need to see that difference." So as an aside, that was something I thought was interesting, that we talked more about data than we did about my pacemaker. So having said that, when we look at this data, as I was doing a training session, I said, well, I don't think I want to be in your medical center in the first quarter of a year. This is 3 years worth of data we took back in the 90s, which is that part's not relevant. So we looked at this data, and I said about the first quarter of the data, and the audience that I was talking to actually started chuckling. And I thought that was an odd response to looking at dispensing of medications. And the response was, well, that's not related to dispensing of medications. And I said, what do you mean? They said, well, and if you look at the lower left of the slide, it says that the only thing that they had to enter, they were able to enter, was the date that they received the report, not the date of the incident. So what this chart was showing, and its bias, was that people were holding back the reports during the fourth quarter because of the holidays, shipping them off in bulk after the first of the year. And since they could only enter the date of the receipt, it biased the data, and it was not telling anything about the dispensing. It was telling about the data collection process. So by not having a field in their data entry screen where they could put in the date of incident, we had a biased result and could actually make a decision here that there's a dispensing problem, when in fact there is no dispensing problem, there is a data collection and entry problem. So this is one thing when we're looking at data through time where we can see bias coming out from the data collection and the data entry issues. We go to the next slide, we'll see another unintended bias put into a system. And this particular situation, when I was doing my training, was the group wanted to look at the incident of patients falling. So these patients were having an issue where if they fell, they found that the rate of falls was something that they wanted to track and see if they could reduce the number of falls. So what you see here is the average line, and then a UCL or upper control limit that says if the data point goes outside this line, we have a signal that something has changed. And you can see from the top, the formula that they used was that the number of falls over the number of patient days or occupancy rate times 1,000. They were looking at fall rates per 1,000 patient days, and that's how they scaled it. And they scaled it that way because that's how they reported the data every month. They had a monthly report. So as you can see across the horizontal axis, these are the data points that we were plotting from the monthly report. Well, the quality assurance people I was working with said, this doesn't tell us anything. That doesn't really give us information that we need to handle the problem. So I stepped back and used my experience in manufacturing, because whenever you do a chart like this in manufacturing, the first thing you look at is what's called a rational subgroup. How are you grouping your data? In this case, we were grouping the data by the calendar. We were grouping it by month, and because we were grouping it by month for reports, we scaled it with that formula at the top. Well, that formula was putting in inadvertent bias. So let's go to the next slide, where I asked them if could they get me the data in actual subgroups of 1,000. So instead of scaling it by the calendar, we used actual values, and we found out that there were actually 21 groups of 1,000 patient days in the real data. And this is the same data plotted with the data grouped by real 1,000 patient day groupings not being scaled. So as we looked at these rates going from left to right, what we discovered was, and this was back in the year of Desert Storm. So I'm an old man and I've been consulting for a long time. The first one on the left, above the UCL line, was a 3-day call-up for many of the nurses that were reservists. There were 3 days for them to leave for Desert Storm, and there was a backfill of nurses, and that chaos showed an increase in patient falls. Then going to the right, the next group, you see a sawtooth there. What we discovered by examining that was that there was indecision on what kind of technique they would use or methodology they would use to try to reduce patient falls. They even went so far as to suggest that patients be restrained, which was really not a popular idea. So when they finally decided on what methodology they were going to use, they had a training event. And so to the far right, we see after the training event, we could see how the training event actually helped reduce the fall rate. And so by having the data properly formatted and properly organized, we got more information and we removed the bias that was unintentionally put in by forcing the data into monthly categories. This technique is also interesting and could be used when we're looking at rates in epidemiology and looking at rates through time, making sure that as we look at data through time and rates, even in epidemiology or other areas, that we make sure we have the data properly grouped to get the maximum amount of information. And then when we go to the next slide, we'll talk about the data techniques. So there's three basic areas that I look at when I'm helping folks look at process data in healthcare. How it's created. Is it coming from a connected device, for example? Is it coming from monitors in an OR? Is it coming from monitors in an ICU? And what about their data entry screens? So the first one, in connected devices, one thing to look at there that I discovered working in, of all places, the utilities industry. If there are smart meters on homes, the data they bring in is thousands of homes in nanoseconds of data. Great for making decisions in real time. What we found was that how that data got stored was not formatted for easy and proper ingestion into analytical software. So the formatting was incorrect and the models did not give correct results because the real-time data was not being stored in a format that was conducive to ingestion into the software. So I got thinking about that. How would that play into if we took historical data from an OR or an ICU and we were storing it? Do we know that it's been formatted properly, that if we try to build predictive models, that it will be properly ingested into the modeling software so that we get the right results? Second group is data handling. Very important as to where and how it's stored. If it's streaming, what are you doing with it? Again, properly formatted. Is it in multiple sources? How do we aggregate it? And most importantly, the thing I hear that's on a must list is, do I have access to the data that I need to make decisions? So a lot of the people who are doing the analysis, do they have access? So we've got to see where we store it, how we format it, and who we give access to, so that number three, can we do the proper analysis we need to do, whether it's trending through time, like I showed you on the two case studies, or then afterwards doing machine learning and predictive analytics. So I've built a Venn diagram on the next slide to try to look at how these three things relate in an organization. So when we're talking about patient records, lab data, etc., collecting and creating that data, that group, again, as Yacine said, it needs to be good data. Then IT or OT, IT being the information technology, OT being operational technology, the difference being IT traditionally handles data at rest, data that's stored. OT is the group that handles data in motion or streaming data from sensors or connected devices. So that's the differentiation there. So then they store, format, and access. And then the third group are the data users looking at doing trending and predicting. The important thing here is that we cannot silo. We must have these three groups working together, talking together to look at what kind of results we want to have. And that intersection of those three is just essential. And we don't want to have any siloing effect of people not working together. So this Venn diagram I put together to show how good decisions are made. And then finally, in the last slide, what I would like to do is talk about something that I learned in graduate school. My degrees are in statistics. And when I got to grad school, our professors emphasized the fact that we should analyze our data before we collect it. When I first heard that, I didn't really understand it. But what they were saying is, what kind of decisions are you trying to make from your data? And what techniques are you going to use to analyze that data? And let's now ensure that we collect the data in the right format, at the right intervals, with the right variables to achieve that goal. So when you talk about that, do you just want to rummage through your data, stratify it, aggregate it, drill down, trend it, etc.? If that's your sole purpose, then how would you collect the data to achieve that goal? If you want to do, and somebody spoke earlier about unsupervised analysis, if you want to go a little bit further with clustering and pattern recognition and those kinds of things, then how would you properly collect the data to get it to go into the software that would give you the ability to answer the questions from that perspective? And then number three, your supervised analysis, where you're trying to predict something, same kind of thing when you're getting into complex analytics. Have you collected the data in a way, as I expressed earlier, I discovered in the utilities industry, and that maps across to streaming data from medical devices, the same kind of thing. Have you got it have you collected it in such a way and formatted it such that it goes into the software and you get the right data in the right format, in the right analytical tool to get the right results? So I thank you for the opportunity to present a different perspective on bias from what I call process statistics, looking at data through time, as it supplements the use of statistics for research and clinical trials and things of that nature.